Welcome to this lesson on S phase, one of the most critical stages in the cell cycle. S phase stands for synthesis phase, and it's when something remarkable happens inside the cell, the complete duplication of all genetic material. Let's explore how this process works and why it's so important for cell division. The S phase, or synthesis phase, is the stage of the cell cycle where DNA replication occurs. During this phase, the cell makes an exact copy of its entire genome. This means every single chromosome gets duplicated. The result is that by the end of S phase, the cell has twice the amount of DNA it started with. This doubling is essential because when the cell eventually divides, each daughter cell needs to receive a complete set of genetic instructions. S phase is part of interphase, the period when the cell is preparing for division. It sits right in the middle of the cell cycle. Before S phase comes G1 phase, or GAP1 phase, where the cell grows and prepares for DNA replication. After S phase comes G2 phase, or GAP2 phase, where the cell continues growing and prepares for mitosis. Finally, the cell enters M phase, or mitosis, where it actually divides. So S phase is sandwiched between the two GAP phases, making it the second stage of interphase. 4 major events occur during S phase. First and most importantly, DNA replication takes place, where the complete chromosomal DNA is duplicated. Second, histone synthesis occurs. Histones are special proteins that help package DNA, and new histones are needed to wrap around the newly copied DNA. Third, centrosome duplication happens. Centrosomes are structures that will later help pull chromosomes apart during cell division, so they need to be copied too. Fourth, checkpoint control is active throughout S phase, constantly monitoring for DNA, damage, and replication errors to ensure everything proceeds correctly. DNA replication happens in 3 main steps. During initiation, an enzyme called DNA helicase unwinds the double helix structure at special sites called replication origins. This creates 2 separate strands that will serve as templates. In the elongation step, DNA polymerase, the star enzyme of replication, synthesizes new complementary strands by adding nucleotides one by one to match the template strands. Finally, during termination, the replication forks where copying is happening eventually meet each other, and another enzyme called DNA ligase seals together any remaining fragments to create 2 complete, identical DNA molecules. 4 key enzymes make DNA replication possible. DNA helicase unwinds and separates the DNA double helix, breaking the bonds between the 2 strands. DNA polymerase is the workhorse enzyme that synthesizes new DNA strands by adding nucleotides in the correct sequence. Primase creates short RNA primers that provide starting points for DNA synthesis, since DNA polymerase cannot start from scratch. Finally, DNA ligase acts like molecular glue, joining together the Okazaki fragments on the lagging strand to create one continuous piece of DNA. Each enzyme has a specific, essential role in the replication process. S phase typically lasts between 6 and 8 hours in rapidly dividing human cells. This makes it the longest phase of interphase. In fact, S phase accounts for approximately 40% of the entire cell cycle in cells that are dividing quickly. The length of S phase makes sense when you consider the enormous task at hand. Copying roughly 3 billion base pairs of DNA with extreme precision takes time. The cell cannot rush this process because accuracy is absolutely critical. The S phase checkpoint is a quality control system that monitors DNA replication throughout the phase. It continuously checks for errors, DNA damage, and incomplete synthesis. If everything is proceeding correctly and replication is complete, the checkpoint allows the cell to pass into G2 phase. However, if problems are detected, such as damaged DNA or replication mistakes, the checkpoint halts progression. The cell then either attempts to repair the damage, or if the damage is too severe, the cell may undergo apoptosis, which is programmed cell death. This checkpoint is crucial for preventing mutations from being passed on to daughter cells.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.